So what we want to do is just go through a bunch of terminology and stuff because we have all these modules for the rest of the day, and we're going to use all these terms, and if no one knows what they are, it's going to be pretty boring, right? So we're just going to try to explain that stuff right now. Just get it all out of the way. We got 30 minutes on my little timer, and we got to get through a lot of stuff. So here we go. So what we're going to do is talk about NGS technologies and terminology. We're going to talk about different experimental designs, whether you have one sample covered to lots of depth or many samples covered to a little bit of depth. You may have, maybe you're only interested in exons, so you do exome capture stuff. There's tons of different designs there. We're going to talk about the data formats for NGS, so things like BAM files and VCF files, and just to let you know what those files look like and so that everyone's on the same page there. And then finally, I'll give a very brief overview of like this sort of general pipeline for doing this stuff, which will be the topic of the rest of today, actually going through that pipeline. Okay. So NGS technology and terminology. So what do we have here? Well, we start with DNA, folks. So we got DNA, and this is like this long thing. It's this 3 billion base pairs. This is a very long thing. And what happens, the very first thing that happens is called fragmentation. So you chop it up into a bunch of random little pieces. So you take this big thing, 3 billion base pairs long, and you chop it up into these tiny little random things. And these tiny things are maybe like 300 base pairs or whatever. They could be different sizes. But they're not anything like 3 billion base pairs. They're much smaller, like 300 base pairs, right? Very small. And they're random, and they're all over the place. They could be overlapping. You don't know where they come from. They're just random little windows into your DNA, right? And then after that, you do this thing called amplification, and you do massively parallel sequencing in the machine, and what you get out is these letters, GTAC, whatever, in a file. Does that make sense? You go from DNA to a bunch of letters on a disk drive. And that's DNA sequencing. And so here, what we're trying to explain here is that not only over the like previous decades or whatever, not only has the machine gotten way more accurate over time, but it's producing tons and tons more data. Like terabytes of data. And I don't mean like 1 terabyte of data, there's like hundreds of terabytes of data. It's a lot of data. And what's really exciting about working in this field or working with the GATK or working at the BroadE is that it's not a matter of like being like more accurate. It's also a matter of being more accurate but being able to deal with hundreds of terabytes of data at the same time. The algorithm, like the things that we do in developing the GATK is not only to improve the accuracy but also to make it work with all this data. And both those challenges have trade-offs, and that's what makes it exciting. Any questions about that one? No? Okay. Okay, so now let's get into a little more detail. So remember I told you about these fragments? So these are these random little collections, random little windows into the DNA. So you put all those things together, and that's called a library. And so you can have, so a library is basically a sample's worth of data, right? Your sample could be sequenced with many libraries, but essentially this library is like, you got your sample, and these are the things that you're going to sequence, and that's like your unit. And then on the other side, we have this flow cell. So this flow cell is like this is like the physical thing that you shove into the Illumina machine and you actually do the sequencing, right? So you can imagine you put some data on this thing, shove it in the machine, and it does its magic. The reason why this thing is important is that you can see you sort of see how the data is organized. And so later when we talk about like modeling the air or covariation of air, you sort of want to know what's like the smallest unit that this air could propagate over, right? And this physical thing is what tells you what that unit is. So you have this flow cell, which is like, several of these lanes put together, and then you have the individual lanes. And so the individual lanes are like the thing where you inject your stuff, and there could be many samples on the lane, and you barcode those samples so you can pull it out later, right? You pull it all together, mix it around, and then those samples are barcoded so you can take it off and you can tell which read or which fragment came from which sample, right? But what ends up happening is you can have errors in the data that's specific to the whole flow cell or specific to only just a lane. And so when you model this error, you want to know about those divisions. So for example, there could be, and this has happened in a project at another center, I won't name names, but you have this flow cell, and then for whatever reason, something stopped in the reaction, right? And then later, the machine operator resumed that reaction, and now every read that came from that flow cell like misincorporated a base at the same cycle. Like cycle 15 has another base misincorporated, right? And every single read that came from just that one flow cell has that error. So knowing it, like modeling those like flow cell specific errors is very important, right? So using the GATK and the fancy error modeling stuff that we'll talk about later, we were able to basically be completely robust to that kind of error because we were capturing that level of covariation of error, right? And so other flow cells like adjacent to it were not affected. It was just this one flow cell that had this problem, right? Pretty catastrophic problem. But the other thing is there can be errors specific to the lane. So maybe someone sneezed on the slide or something, and that sneeze molecules just hit one of the lanes, right? And so now you have errors in just that one lane that's not affecting the other lanes. And so building an individual error model per lane is how you're able to capture that kind of variation. Does that make sense? The divisions there? Okay. So the actual physical device as part of the sequencing is what gives rise to the divisions of the error, to the covariation of the error. So that's pretty important. Okay, so after you've done the sequencing, now you have to actually do something with this data, right? And so the first thing that happens is you take all these reads, and a read is just, we're reading off this fragment, and we have, one little snippet of DNA. We're a read of this fragment, right? And we have a pile of them. We have billions of these things, right? And they're tiny, and they're from all over the genome. And the first thing we do is we map it to the reference. So the human genome reference is this, humongous long thing, and you want to take all these little reads and find out where it goes on the reference. So you want to put them somewhere. And you don't really care about their exact placement, to be honest. Like, you just want it to be close. You want it to be in the ballpark. This read was supposed to go somewhere here. And this is, of course, a very computationally intensive step because you have this huge genome and you have these tiny little reads and you have to search the whole genome to figure out where it goes. So it's crazy computation. But that's basically, and we'll talk more, we're going to go through all these things in detail more. I just want to introduce the topic now. Does this make sense to anyone? Cool. Yep, I can tell that you've all had this material a long time. I hope someone's getting something out of this. Okay, so you can take those aligned reads, and now you can view them, right? And so we have this great thing developed at the BroadE called IGV, the Integrative Genomics Viewer. It's developed by Jim Robinson and his colleagues just down the hall from me, and it's beautiful. So you load in this data, these sequence reads on disk. They've been aligned, and you load them up, and this is what you get. And so all these vertical lines here. The horizontal lines. All these horizontal lines here are the reads. They're like the little things that you read off, and they're just laying there, right? And when they're gray, what that's saying is that those bases match the reference. There's no reason to worry about them because they just match the reference. And you have the reference down on the bottom. There's all these little letters, A, C, T, and G. They're colored for you so you can pick them out. And they're just there, and they all match, and that's great. And what you see in the middle here is what a beautiful heterozygous SNP looks like, where half of your reads are showing the reference allele, and that's why they're gray, and the other half of the reads are showing the alternate allele, and that's why it's colored as a T. So the reference in this location is a C, and half the reads are showing a T, so this is a SNP, a beautiful heterozygous SNP, very easy to call. Right. And so what else are we showing here? Oh, so the other thing to say is that when you have these fragments, what you're going to do is it's called what's called pair end reading. So you read from one end of the fragment and then you read from the other end of the fragment. And what you see highlighted in purple here is both of those reads. So you have two reads of the same fragment, and they happen to be overlapping here. So they both have the T, and they overlap at some place. And you can, in IGV, just click on one thing, and it'll show you where its other mate pair is, where its other fragment is, other the other read that came from the same fragment. And so that's a beautiful way to view things. You can see that, I don't know if you can really see, but there's little arrow markers here. So this read starts here and reads in this direction. And there's a little arrow marker here. So this read was way over there, and it read in this direction. So that's cool to know which way you're reading. And the other thing is that you probably can't see it at all, but there's really grayed out bases there. Like this is a C. It doesn't match the reference. So it's a C, but it's very light. And so what it's trying to indicate there is that the machine read a C, but it wasn't very confident in itself. So maybe it's a C, maybe it's not, who knows. But it's trying to let you know that it didn't know whether it was a C or not. So it does a lot of beautiful stuff. Hopefully you will all download this right away and start using it. I use it every single day, many hours every single day, and I love it. Okay, so that's really it about NGS. So if anyone has a burning question about DNA or something, now's the time to ask it. Okay? Okay, so let me just take a moment to check the time, because I'm famous for just rambling on. Okay, we're doing well. Okay, so now we're going to talk about experimental designs. And so the thing here to think about is like you have a fixed sequencing budget, right? Someone gave you a grant, you have some money, you want to do something, you want to do some science. And so the basic trade-off here is you can have fewer samples that are covered to lots of depth, and that's on the left side, or you can have more samples, but they're covered to less depth. So maybe you have, so that would be an example like the 1,000 Genomes project, which has 2,500 samples, but each sample was only covered to 4x depth. So, and the trade-offs are interesting. So on the left side, you're pretty confident in the variation that's in that one particular sample, right? You're able to find basically all the variation that one sample has. But if you're interested in like human genetic variation, that's a pretty small fraction of the human genetic variation, right? So on the other side, you find way more variation is discovered in your population, but your confidence in any particular person or any particular sample or any particular genotype is way lower, right? Because in this example, you only have 2 reads at each sample, so you have no idea, just based on random sampling, whether you found this person's variant or whether maybe they're heterozygous, but the 2 reads that you happen to see held the variant, so now you think it's homozygous. So your ability to genotype when you have fewer reads goes way, way down. But if you're not worried about the particular genotype for a particular sample, then this is the way to sort of sample the space of variation. Does everyone understand those trade-offs? Okay. It's pretty cool stuff, actually. Yeah, anyway, we'll keep going. So here I'm showing, so now we're going to go through 3 designs, and we'll talk about the trade-offs in more detail. So here we're showing high-pass sequencing design. And so this is whole genome coverage, so that's why you have a bunch of reads that are way off in the hinterlands and the introns or intergenic regions are in the middle of nowhere. So the whole genome is covered, right? So this is for like a single sample. So you have 3 billion bases that were covered. And in general, this is going to work out to be 30x coverage across the genome for 8 lanes of HiSeq. I don't know if those numbers are current anymore, but we'll just pretend they are. And so what you're going to find when you do this is you're going to find about 3 million or more variants in your sample, right? Like 3.3 million SNPs and, I don't know, 300,000 indels or something. So that's like 4 million. And you're going to find near 100% of the variation in this sample, right? You're going to find basically everything because we're pretty good. What else are we saying here? Right. So let's contrast that with low pass. So in low pass, now we only have 4x of the reads, right? And because of this, so your coverage is like, your average coverage is like 4x, and this is maybe 1 lane of HiSeq. But your ability to find all the variation in this sample has gone way down. Now you're at like 90% of the variation, because a bunch of it you just missed due to random sampling. You know, with 4 reads, maybe you just accidentally, the read with the variant didn't show up, right? So that's happening in the middle here. So it was covered by 3 reads, and it was a heterozygous SNP, so that the read with the variant just never showed up due to random sampling. And so you missed it. It was gone. That's basically it. Okay, and now let's contrast it with exome capture. So maybe you're only worried about the exons, so you do this fancy thing called exome capture, and now you have reads that are in basically the exons and the flanking regions. And so now you're targeting a much smaller region. It's like 30 megabases, right? Much smaller than 3 billion bases. The way this process works is the goal is to get 80% of the exon targets to be covered by 20x, 20 reads or more. But what ends up happening in practice is that the majority of the exome is covered to like 100x. It's just some of them are very hard to capture, and so you have to like bump that way up in order to capture those hard to capture ones. Well, so these numbers are for, the idea is it's supposed to be 1 human sample. So like, you know, the exome in some other organism is going to be a different size, for example, and the amount of variance that you find is going to be different, things like that. So, but the idea is that really the difference between, so we're just really trying to show you the relative difference. So the difference between 3 billion and 32 million is humongous, first of all. And also the difference between finding 3 million variants all over the place versus only finding 20,000 variants, very different, right? So you just have, it's much fewer things to actually go hunting for. So maybe you're doing like a disease study, and maybe you think that the cause of your disease is in the exome for whatever crazy reason. And so you're just narrowing down your search space to something much smaller, right? But what you're doing is, by doing that, you're losing all the variation in the genome. You're only going to find 0.5% of the variation across the whole genome by doing this. But the ones that you do find, perhaps they have special relevance because they're in the exome. That's the basic trade-off. And so what it looks like is in the middle, there were real variants like in the intron and all this other stuff, but you just have nothing there at all. It's just blank. It's just a sea of blank. But then when you get to the exon, now you have lots of coverage. So your ability to genotype in those areas is great. You just have no ability to call variation off of the targeted region. That's the basic trade-off. Any other questions? Thank you for the question, by the way. It livened everyone up, I'm sure.
The fact that you have 95% of the single-node discovery means that most of the single nodes are in the exome regions, or this is 95% of the single nodes that are in the exome?
Yeah, it's 95% of the single nodes in the exome. Sorry about that. Yeah, because there's tons of variation that you're just not calling at all. You're not even worried about calling. Yeah, so that's 95% in the exome. Everyone good there? Okay. So now let's talk about data formats. So this will be exciting, I'm sure. So, so what we have is, so we have this machine, right? And that was the machine where we stuck the slide in, it did its thing, and we wrote this thing to disk. And the thing that we wrote to disk is the FASTQ file. So inside that machine is this thing called the base caller. And so this is hey Eric, this is this thing that you know, it's specific to the company. They have their own thing. They look at their internals, like their laser beams and all this stuff, and they say, oh, I found an A, or oh, this is definitely a T. This was a C. And that's how you go from the DNA to the fragment to writing it out to disk, right? So this is this proprietary thing inside the machine called the base caller, and all it does is it's spitting out letters, A, C, T, G, whatever, whatever, and you're writing that to disk, and that's in the FASTQ file. And so we're going to go through all these files, and I'll show you what they look like, but here's just the name of them. So after that, you do this alignment step that we talked about, right? You take all those little pieces, you put them on this humongous genome, and that's called an aligned file, and that's the SAM or BAM format. So SAM is this thing, and BAM is just the binary version of it. And so those are aligned reads, is just a BAM file. And then after that, you use something like the GATK or whatever you want, and you make variant calls. And then that format's called the VCF, variant call format file, and that's just a list of genetic variants. So these are the three like basic file types in NGS processing, and we're going to go through each one, and I'll show you what they look like. But these are like the three things you gotta. These are pretty important, actually, if you want to do this kind of stuff. Okay. So the first one, FASTQ file. So the FASTQ file is just the raw unaligned reads as it came off the machine, and each record is just 4 lines in the file. It's very simple. So the first line is a unique identifier supplied by the machine. It's going to have this @ sign and just a bunch of random stuff, and that's interpreted by the machine. It's just a random identifier for every read that come off the machine. So there's going to be billions of these things. There's going to be billions of unique identifiers. So the next thing is the actual sequence that the machine read. And for this, it's like a 20 base pair read, and the machine happened to read a bunch of C's and T's, and this is what it is. Right? So just spit out the letters right in a row. The next thing is just a plus sign. It's pretty simple. So it's just a plus sign. Every time, a plus sign. And then after that, you have another string, which is the string of quality scores. And that string is going to be the same length as the string as the base sequence. And this is a fancy encoding, which is it's ASCII plus 33, right? So 33 is like the first printable character in ASCII. So you take a number, add 33 to it, and that's what you get right there in ASCII. And so this is a special encoding to tell you, so a base quality score is like telling you how confident the machine was in the base call. So maybe it called a C, but it was only 95% confident. And then it called the next C, and it was like 99.99% confident, right? And so you encode that probability using this special thing called Phred scale into just a single character, and then you put that in the thing there. And on the next slide, I'm going to explain what Phred scaling is, so don't worry quite yet. But does everyone see how this is a string of bases and then a string of probabilities right underneath it? It may not look like probabilities, but all it is just a probability. Good? Okay, and so you'll have a billion of things in the record, and so your file size will be a billion times 4, right? It'll be the number of reads times 4, because there's 4 lines per read. That'll be the size of your file. It's just an ASCII file. You can cat it, you can grep it, you can do anything you want to it. And that's it. Mm-hmm. Yeah, so what's going to happen is like this first semicolon corresponds to this C. I don't know what the encoding is, but we'll just pretend that that's like Q20, right? So this is you're saying you're 99% confident in this C. And then the next C is also 99% confident. And this C, maybe a 3 is worse. We'll just pretend that that's like 50% confident, right? So you're very confident, but then this C, it was not very confident. And so you just go, it's just one character per every base in this sequence. So it matches 1 to 1. It doesn't look like it because of the formatting, but if we use what is it called, a sans serif font or whatever it's called, then they would match up completely. Fixed width font. If we used a fixed width font, it would match up exactly, and you would be able to see that one corresponds to the other. And it's just a probability. Someone else?
What is the maximum Phred allowed?
Well, it's the maximum ASCII number. So the file format's like, it's like the ASCII, the printable characters goes from 33 to like 100 something. And so the maximum Phred is like 96 or whatever that difference is. It's just the it's just whatever's in the spec to say what the maximum printable character is. You know what I mean? It's not so important because a Phred scale of 90 something is like crazy, right? That translates into a probability that's like crazy, and so astronomically high, right? So you would never have that much confidence in a base, and if someone said they had that much confidence, you shouldn't believe them, right? So I'll explain why it's crazy in a minute, but you'll see that a Q score of like Q90 is crazy. That would be very, very confident, right? And no one is that confident. They shouldn't be. Does that make sense? Yeah. Okay, so let's talk about Phred scaling. Let me explain what these things actually are. So all it is, the mathematics is it's -10 times log 10 of the error rate, right? So let's go through some examples. So Q10 is 90% confident, so you have a 10% error rate. And then it's a logarithmic scale. So Q20 is like 99% confident, or a 1% error rate. Q30, 99.9, or a 0.1% error rate. And it just goes down logarithmically like that, right? So when I was telling you about the ASCII encoding, you're taking like number 10, adding 33 to it, turning it into an ASCII character, and that's the encoding for the probability, right? And so you can see that a Q90 would be, you can work it out as point whatever, 0.1 error rate, which is quite low and much, much lower than the error rate of any particular machine. Does that make sense? Okay, so after the FASTQ file comes the BAM file, or the SAM. And so BAM is just the binary version of SAM. And so, you know, back in the olden days, we had all these companies, and they had all their own sequencing formats, and it was a nightmare. I have to tell you, it was crazy. Like, trying to deal with 454's BAM, or 454's file versus Illumina's file versus all these other companies' files was crazy. So, eventually, everyone converged on this beautiful file format called the BAM file. And all it is just those same reads that we saw in the FASTQ file, but now they're aligned to the genome. And so because of that, you have some extra fields in the file. So this has been turned from the binary version into ASCII, and then now I'm displaying you one record at a time. And so it starts with the read name. And so this is the same unique identifier that was given to it in the FASTQ file, right? It's the same. It's just a unique identifier. You have the location on the genome now because now it's aligned to the genome. So now you have chromosome 1 and this crazy position somewhere in chromosome 1, right? So this read is actually, now you know where it's physically located. It's not just a random fragment of the genome anymore. It's now been aligned to the genome and you have a location. And the next thing, the next field, is called the cigar string, and that's how you know how that read is aligned to the genome. So whether it has gaps in it, whether there's insertions or deletions, So what this is saying is that this read is aligned 23M, so 23 bases are aligned, then there's a gap of 6 bases, 6N, and then 10 bases align after that. So 23 bases align, a gap, and then 10 more bases align. So this is letting you know whether the aligner thought that like certain parts of the sequence were inserted or deleted, and it lets you know how it's aligned to the genome. Okay? So after that is the read sequence. Now you have the DNA. You have the same quality score in the same format, the same scaling, and then you have a bunch of metadata that's very important. So it is the Phred quality. It's the base. It's the base. Yeah, it's still the base quality. What about the aligner? So there's a bunch of metadata, tons of metadata actually, and one of those is called the mapping quality score, and that's a score that's assigned by the aligner to tell you how well it was aligned. That's also a quality score assigned by the aligner. So there are tons of like metadata fields that an aligner could populate to tell you more information about this read. The one that we're showing here, though, is very important because it tells you which lane the data came from, right? Because remember I told you knowing about the lane, like knowing about the specific errors per lane is very important. So now you have to associate every read with which lane it came from, right? And so there's tons of metadata in there to let you know where it came from. So you know which error model to apply to this read because you know that it came from a certain lane that had a certain error. What's up on the back? I didn't hear anything because my alarm just went off telling me that I've gone way over time. So please continue. Well, so that's a good question. I think what happens is in the FASTQ file, they're going to be assigned the same identifier, so you know that they came from the same fragment, right? It's a fragment identifier. And then in here, what's going to happen is that there's like data that says I'm a paired read, and my mate's pair has this name. So they're like associated with each other in the file, right? In this metadata, there's tons of stuff in there to tell you where your mate is, where your buddy is, right?
For the FASTQ, usually for the FASTQ, it's two different. If it's pairs, then it's two different FASTQs that are just matched. Like the first one here is the matching case.
Okay. Well, the question was about mate pairs, and how do we know where these mate pairs?
One next to each other.
Oh, in the FASTQ file.
Yeah, in the FASTQ, like
Okay, so in the FASTQ file, they're like in sequence. They're like bam, 1, 2, 1, 2, 1, 2. They're just in sequence.
Like that, there's actually two different FASTQ.
yeah, two different FASTQ.
We are one FASTQ and we are two FASTQ files. They have the same read names, same IDs.
Okay.
And obviously they use the
All right, so you guys know this stuff way more than I do, but cool.
Usually it's two FASTQ.
Good? What's up?
Is there a universal structure or way data is recorded in the metadata section, or does every single liner record it?
No, so there is a spec. There's this thing called the BAM spec, and it specifies lots of things. Like we asked about this alignment score, right? And so that's part of the spec. There's tons of other things about this read, about the mate pairs and stuff that are part of the spec, and so that's the same for every aligner. But there's lots of stuff that some aligners do that others don't, and they add those as these tags, as these metadata tags, and those are all different depending on the aligner. So like some aligners really care about structural variation, so they'll tag reads that look like they come from structural variants, you know, like special things, right? And so the answer is it's both, but the majority of things that are important are common amongst all of them because it's part of the spec. Of course, if you wanted to write your own aligner and you had some fancy idea for what was important about the read, then you could encode that just by adding a tag to the read, and it would be your own tag, right? And no one else would know what to do with it, but at least you would know what to do with it. So it's extensible in that way, which is nice. Unfortunately, we're going to have to zoom along because I'm way over time. So let me just say that there's, in addition to having all these records in the BAM file, there's the BAM header, and in the header, the most important thing is that you're gonna find out what all these lanes were, and these are called read groups. So we have this first RG is a read group, and that's the lane's name, and then it tells you somewhere. It tells you which sample is in that read group, right? And so that's how you're going to associate. That's how you're going to associate every read with whatever error model you generated per lane. And so being able to have this like lookup table in the header file is what allows you to do that sort of thing. And so that's very important. There's also tons of useful things about it lets you know like this file was processed with BWA and this is the version number, and then it was processed with the GATK and this is the version number. So there's tons of like really important information there if you care about how the history of where this file came from, right? So let's zoom along, and the last one we have to talk about is the VCF file. So the VCF file, it's the list of variants, right? And so I'm just going to go very quickly. The most important line here is we have this header line, which is chromosome position ID reference alternate. Hopefully you can see that. And then we have all these records, right? Each one is a genetic variant, right? So this one's at chromosome 20. It has this position. It has this ID in the database called dbSNP, or you can put whatever ID there you want. And what it's saying is that it was a G reference allele and an A alternate allele. So this was a G to A SNP. It has, and then after that, you have these three samples. NA is 1, 2, and 3. So these are three samples that you processed, and those samples, you can ask, what are the genotypes for this variant for those samples, right? So this G to A variant, the first sample was a 0, 0, so that means it was reference. So this is the 0 allele, and this is the 1 allele, so it's a 0-based indexing. So this person was reference, and then but the next person was heterozygous, because it was a 1, the A allele, and a 0, which is the G allele, right? So the next sample was determined to be heterozygous for this variant. And the final sample was homozygous because it's a 1, 1. So both of the chromosomes had an A. Make sense? It gets way more complicated than that if you want it to. So you can have multi-allelic variants. So this variant is at this position, the next line down, this position. And the reference allele is an A, but it's saying some people in the population were G and some people were T, right? And so this sample, the NA1 sample, is a 1, 2, so meaning that one of the alleles was G and the other allele was T, because it's a zero-based indexing, right? So GT, this middle sample was also GT, and the final sample was TT, homozygous for the T. Does that make sense, how it can happen? And you can have indels, you can have lots of things. So you can have, like this would be an indel, right? This GTCT goes to G. So this TCT was deleted, right? And so the way the formatting works is that there's always an anchoring base so that you know where it goes, and then the TCT is deleted. So maybe this sample, 0/1, is heterozygous for that deletion. Make sense? So this third field is the ID. And so whoever made this variant call thought that they would like to call it microsatellite number 1, right? It's just a unique identifier, or not even unique, it's just an identifier. For whatever reason, whoever generated this file thought that that was a good name for this variant. Most of the time, you're going to find that it's just a dot, meaning there's no ID. Sometimes those are in a database like dbSNP, and so it makes sense to put the dbSNP ID there. That's just an easy way to reference back to that big database. But you can put whatever you want there. My favorite variant number 1, my beautiful variant number 2, whatever you want to put there, you can put there.
Yeah, the nomenclature, why sometimes pipe, why // what's different?
Yeah, so that is you'll find out about that stuff, I think, in the tomorrow session. So that's called phasing information. So what it's saying is that, just briefly, what it's saying is that it knows which chromosome these came from, right? Like it knows that this 1 and this 0 go together. They were on the same haplotype. And this 2 and this 0 go together, and they were on the same haplotype. And so whoever generated this file had confidence in that phasing information, and so they were able to say which haplotype actually these variants. So it's a way to connect information from one line to another, whereas the slash is just, we don't know, each variant's independent. Okay? I've taken up way too much time, so let me just say that this is, the sequencing companies might want you to think that you could take your DNA and go straight to medicine, but what the point of this whole workshop is that you take the DNA and you sequence it, but in order to go to actual variation in medicine and do these cool things, you have to do a ton of work to actually call this variation and find out what's real and what's not. And that's sort of the topic of this workshop, and that's why you use the GATK, is to do that. So what it looks like is, I showed you that very beautiful IGV screenshot where it was obvious that there was a heterozygous SNP in the middle, but in fact the data looks like this, where it's messy all over the place, and you have to use very sophisticated statistical tools to decide when the variation is real and when it's not. And that's what we spend all of our time doing, is trying to develop those algorithms, these statistically robust algorithms, to decide in the face of this messy data where the variation is and if it's real or not, or if it's just an error, and trying to assign accurate probabilities to these variants and all that sort of stuff. So I think that is it. So I hope you have fun over the course of this workshop, and thank you for your attention.